Thank you. So welcome to the talk about Foldseek, our ultra-fast similarity searching and clustering tool for protein structures. And this is a very nice and very enjoyable and fruitful collaboration with the group of Martin Steinegger at Seoul National University. And yeah, this is our team. So Michael from my group and Stefanie from Martin's group are first authors here. So it was already a very nice introduction by Sebastian why we have a challenge coping with the deluge of data that is now coming to us from AlphaFold-2 predictions. So I think you probably all agree in this auditorium that AlphaFold-2 is really revolutionizing protein bioinformatics and structural protein structure. So Mohamed Al-Quraishi said about a bit more than a year ago, everything we did with protein structures, with protein sequences, we can now do with protein structures. And this was the time when we started thinking what we can do to cope with the amount of data that we were expecting. So the announcement is that by the end of the year, I think now they've shifted it to early next year, EMBL EBI in collaboration with DeepMind will make available over 100 million protein structures that are basically at almost the level of accuracy of experimental structures. So these structures are of course extremely valuable, but they are only as much valuable as you can really do something with them, and because it's such a huge amount of data you need, for example, to be able to search them quickly. And this is of course the topic that we are addressing. So why could this be useful? For example, for annotation, if you have sequences like this one ubiquitin and the small ubiquitin-like modifier sumo, they share a sequence identity of 16%. Despite the fact that they are actually very similar in structure. So after more than 1 billion years ago, they still look very, very similar in structure, and they actually still perform a similar function. So we need structure comparison to be able to, for example, use the AlphaFold database that we'll have with these 100 million structures, for example, for annotation, of course, also for many other things. So the problem is that the current tools are actually really much too slow to do exhaustive searches. So we did a test with the 800,000 structure containing AlphaFold database, searching it with an RNA-dependent RNA polymerase of SARS-CoV-2. And this took 1 week with DALI, a bit more than a day with TMalign. And if you extrapolate that to the 100 million structures we'll soon have, then this means that even TMalign would take half a year on a single CPU core, which is of course not super practical in the long run. And Foldseek, which I'm going to explain to you now, is taking 5 seconds for this search, which is 4 to 5 orders of magnitude faster. But this is actually rather an underestimate because it scales much better than these tools. These tools will probably you can be lucky if they scale linearly with the size of the databases, whereas Foldseek, we expect it to be scaling sublinearly. So we built for how do we gain the speed? We built on a actually 2.5 decades old idea of reducing structure comparison to sequence comparison. So the idea is that every residue in your protein structure is represented by some structural state in a structural state alphabet, and then you can just write down the sequence of structure states that would represent the protein, and then do comparisons between these sequences. So this idea has, as I said, has been proposed quite a time ago, and since then many of these alphabets have been proposed, and they have one thing in common: they all describe the backbone of the protein. They are basically refinements of the secondary structure alphabet with just 3 states. So if you then have a protein, you can translate it into one of these alphabets, for example, the CLE alphabet you see on the left. And then every structure is just a sequence, and then you can learn the substitution matrix between these structural states from structural alignments in the same way that you learn Blazon 62 from sequence alignments. And this then effectively reduces structural comparison to sequence comparison, obviously makes it very fast. The reason why these tools have not become much more popular despite their speed is that these tools are not nearly as sensitive as true structural aligners like TMalign or Dali. And the reason is that they don't really capture much information about the structure. So most of the information is, of course, in the cores, and where you have mostly secondary structure elements like helices and strands. And these helices and strands are really mostly described, mostly described in such rather uninformative stretches of H's or sheet-like states, whereas the most information is expanded in regions, in loops, where you actually have the least conservation. Where, for example, these 9 coil-like states are very detailed, but often used in loops where they're not really conserved. And on top, you have this very strong dependence of residues on each other, and neighboring residues on each other, which reduces the total information. So the idea in Foldseek was to not describe backbone, but to describe tertiary structure. And what I mean by that is that for every residue that we want to encode now in our letter, we search for the residue that is nearest in 3D space, and then we describe the conformation between those two, meaning we describe the little piece of backbone of each of those. So the the 3 C alpha atoms around the red one and the 3 C alpha atoms around the blue one. And the conformation of these 9 residues is described with a 10-dimensional descriptor containing angles and distances. And these 10-dimensional vectors are then discretized into 20 letters. You could do just a clustering, k-means clustering, for example. This is actually what we started with, but you can do it in a much smarter way. And this is what I will explain to you now. So the first thing is that you don't just take as a nearest neighbor, the residue that has the smallest C beta distance, C beta distance, but you use so-called virtual center, what we name virtual center. And I will explain on the next slide why this is important. So you find your nearest neighbor for your red one, then you extract this 10-dimensional descriptor, and then you need to have an algorithm that can give you the state for this 10-dimensional descriptor, and this is an encoder. And so this is a neural network of I think 3 layers we use, and the result is one of those 20 letters. And crucially now we train this encoder as part of a variational autoencoder. So you try to predict what is on the right side by what comes in on the left side, and fiddling it through this information bottleneck in the middle, the 20 states. And very importantly, what is on the right side is not the same as what is on the left side, like in a standard variational autoencoder, but it's basically the homologous feature in a structurally aligned protein. So you try to predict the TD descriptor, 10D descriptor of the aligned part from a structural alignment. So this is how, and then we train the autoencoder on these training alignments and get out our encoder, which now embodies our alphabet. So why do we need these virtual centers? Well, for 2 reasons. We want to maximize the conservation of interactions, so of the nearest neighbor interactions. And we want to be sure that in a structural alignment, in a homologous structure, what is nearest neighbor in one should be the nearest neighbor also in the other, as often as possible. And this can be done by basically moving away from the C beta and moving it somewhere else in space, given by fixed conformation to the backbone. And the second source of gain comes from making sure that we don't get that we mostly get tertiary interactions. So we describe interactions that are far in sequence, and as few as possible they are local, because those local interactions will lead again to the same problem that we have with the backbone conformations. We will have a lot of repeated letters, and this reduces the information, the total information that we have when comparing sequences, from the naive one that is just letter to letter, between the 2 sequences, to a reduced mutual information that is corrected by the fact that we have redundancy between the letters in one sequence. So what we actually find now is that when we look at our alphabet, the corrected information that it takes into account this correlation between neighboring letters is actually much, much higher than in CLE and also, of course, in the amino acid alphabet. So the 3D states, just to give you an overview, look like this. The 20 that we have defined. So they are pairs of little pieces of 3 C alphas, some in beta-beta conformation and many in many other conformations. So coming back to our example, the ubiquitin sumo alignment, this is the sequence alignment, 16% sequence identity. This is the Foldseek 3D alignment. Now you see the sequence identity is 54%. So you see that these states are much more highly conserved than the amino acids. Now, how do we gain additional speed? We have now a sequence-sequence comparison method, but we want to, of course, be much faster than standard Smith-Waterman. So we use modules from our MMSeq2 library, which is strongly optimized for very fast comparison. And it uses these MMSeqs and Foldseek use 2 pre-filters that are crucial for gaining these high speeds. The first filter looks for 2 6-mer similar k-mers, so 6-mers, in the same diagonal. So this is what is shown here in black. And only those targets that have one of those, that have 2 similar 6-mers, will get looked at in the second step. And the second prefilter will then calculate the optimum gapless alignment for each of those diagonals where we found these two similar 6-mers. And when this score is above a certain threshold, then we will only do the full Smith-Waterman alignment using the 3DI sequences. And all this is of course, implemented with AVX2 instructions for maximum speed and a 32-fold parallelization on a single core. So to look at now performance, I think what our goal was from the beginning is to be as sensitive as possible, and we actually reached sensitivities that are similar. So in finding homologous structures that are similar to the most popular protein structure aligners, DALI, TMalign, and CE. So you see here Foldseek in red and the other structure aligners in gray, blue, and green. And what you see here is this, for every query in this all against all comparison of the SCOP database. So SCOP is a single domain database where we have a hierarchy of family, superfamily, fold, and so on. So for every query, we have a sensitivity. The sensitivity just tells us how many of the same family we found before the first false positive. And now we have a cumulative distribution over the sensitivities for each of the queries. And you see in here, DALI is more sensitive than Foldseek, which is as sensitive as TMalign and a bit more sensitive than CE, whereas the CLE library is here much less sensitive, and even less sensitive is simple sequence aligner MMSeqs. And on a superfamily level, this looks relatively similar, only with a bit less performance. Because we wanted to also have a benchmark that can compare full-length sequences, full-length protein structure models. We use the AlphaFold database, and I cannot go into the details, but this is a reference-free database where we use LDDT scores to decide whether a residue is in a part of an alignment that is structurally correct or not. And again, we see that Foldseek has similar sensitivity to discover correct alignments as TMalign and Dali, so that you can see here these 3 cluster relatively well together. So high is good. Whereas CLE, this backbone alphabet, and MMSeqs are much below. And in terms of alignment quality, Foldseek is actually achieves the highest sensitivity, so the most residues correctly aligned, and is only less precise than CE. Yeah, and another advantage of the Foldseek algorithm is that it basically doesn't make bad mistakes. So a bad mistake is made here by TMalign, and the reason is that these mistakes happen is that they use a lot of heuristics to make sure that they have a good chance of finding structurally superposable targets. And this is a bit a game of luck. They make sure that most of the time they find the right hits, but it's by far not always the case. And in this case, the query is this white structure here, and the best hit they find in the SCOP database is the red one, and this is not even in the same SCOP class. Whereas dynamic programming always will give you the alignment that scores best, and usually this is very robust. And if there's something that you can find, then usually that is within your search sensitivity, you will find it. So the Foldseek here finds the correct family. Now, I just want to mention briefly that we have a web server where you can directly try the Foldseek algorithm out. So it's at search.foldseek.com. You paste your PDB sequence, your structure in, you get a list of results. You can click on any of the results, show the sequence alignment, show the structure. You can highlight parts of the structure in the alignment. So you see here now the highlighted part in red. You can look at the alignment, the aligned residues with this little arrow. You can look at the full structure of the query with this gray button. You can look at the full structure of the template in red. You can go to the full screen view and so on. So this was programmed by Cameron Gilchrist. Yeah, and as a summary, so Foldseek is a new algorithm that encodes the structure of proteins as sequences that are discretized by tertiary interactions. Can search billions of structures in minutes, 4 to 5 orders of magnitude faster than popular aligners. This has comparable sensitivity and alignment quality. Actually has extremely accurate e-values in contrast to structural aligners. So you see here the dotted line is on top of the red dots. So it's very, this is of course very useful for annotations. It produces local alignments, not like structural aligners that mostly produce global alignments. It's robust against relative movement of domains, and it's already quite widely used. So more than 100 downloads per week and more than 1,000 repo views per week. And with this, I finish. I thank you for your attention. I hope that you now agree with me that what we did with sequences, we can now do with structures. Thanks. I don't hear anybody moderating. Should I just start answering questions in the chat? Maybe I'll just do that. So I will just pick a question here. How does the...
Sorry, I was muted. That is the question. It's posted. I'm sorry. Go for
yeah,
it.
so should I just read it?
Yes, sure.
Yeah. How does the cardinality of the new alphabet affect speed and alignment quality? So short answer, it's in the supplementals of the bioRxiv. There's a bioRxiv out, and you can look it up. You can get better with 40, for example, instead of 20, you get slightly better. But on the other hand, it actually has a large impact on speed because we need to find these similar k-mergers. You remember in this very fast prefilter. And this similar k-mergers means we're not looking at exact matches only. We're looking at all the similar ones. And and basically this explodes. If the alphabet gets larger, then the number of similar ones that we have to generate explodes because you have to have alphabet to the power of k proportionality factor there.
Are there any questions from the house? Yeah, sure. There's a question, can you align two user input structures?
Actually, that's a very good question. I think not yet, but this is easy, of course, to do. On the other hand, that you might actually also, of course, do with TMalign and Dali. So the point, of course, of the server is that it is very fast. But it certainly might be useful. So I think we should add it.
Yeah, another question. So why an alphabet of 20? Have you tried smaller, bigger? Is it an optimum of some sort?
Yeah, it is a sweet spot. It is a bit by chance that it is also the same size as an amino acid alphabet. I mean, we chose it, of course, since we saw it was in the right range, we put it at 20. If you make it too large, as I said just now, it becomes hard to generate all the similar 6-mers. If you make it too small, you lose a lot. So basically, you gain logarithmically. That means if you go from 20 to 10, you lose similarly much as if you gain from going from 20 to 40. So it is kind of a sweet spot.